I'd like to welcome you all back to the Environmental Influences on Brain Health that's going to be moderated by Dr. Cheryl Walker. She's the director of the Center for Precision Environmental Health at Baylor College of Medicine. And Dr. Walker is also a member of our 2019 conference program committee, so we're very grateful for her and the time that she's helped us with in putting the planning together for this conference and this great panel. So Cheryl, it's all yours. Thank you.
Thank you very much, and I'm so delighted that the Academy, and especially the other members of the program committee, responded immediately and positively when I suggested that we should actually look at the influences of the environment on brain health. And we've had just incredible talks that have focused on the genetic side of the house, and I've certainly learned a lot. It's been fantastic. And so what you're going to hear about today is really some of the best research and frankly, some of the most exciting research being done in the environmental health sciences across the board at looking at how environmental exposures affect the brain. So we have a stellar lineup, and we'll go ahead and get started with Dr. Andrea Gore. She's a professor and the Vacek Chair in Pharmacology at UT Austin. She just stepped down as editor-in-chief of the journal Endocrinology. This is the lead journal for the Endocrine Society. She was also the lead author on 2 position papers that the Society has put out about the potential health effects of exposures to endocrine disrupting chemicals. She's a fellow of the American Association for Advancement of Science. She's also a recipient of some of the Endocrine Society's highest awards, including the Laureate Award for Outstanding Public Service and the Edith Clark Women of Excellence Award. She's going to talk to us today about the effects of environmental exposures on the brain, and particularly on the epigenome, and the effects of those epigenetic effects not only on the exposed generation, but also on subsequent generations. So, Andrea, thank you so much for coming today.
Thank you, Cheryl, and I'd like to thank you and the organizers for inviting me and for including a session on the environment to remind everybody that the environment does in fact play a really important role in the brain, and particularly in brain development. So let's see. Everybody had trouble with forward and back, so there, okay. So I'm going to talk today about a group of chemicals that are called endocrine disrupting chemicals. I'll refer to them as EDCs. And there are hundreds of thousands of chemicals that have been synthesized. Many of them, probably the majority of them, are somewhere in the environment or are in use from having been synthesized. And of those, about 1,000 have been tested and have been shown to be EDCs. Many more may be EDCs, however, they probably just haven't been tested yet. And a group from the Endocrine Society that I worked with defined an EDC as an exogenous chemical or a mixture of chemicals that interferes with any aspect of hormone action. We are exposed to EDCs through a number of sources, a subset of which are shown here. I'm going to be talking today about 2 types of chemicals. One of them is the industrial chemical group PCBs, polychlorinated biphenyls, and the other is a fungicide that's called Vinclozolin. There's evidence that every human has EDCs in our body. The CDC has done a lot of testing. It's found in body tissues, in urine, in serum, in umbilical cord blood, in amniotic fluid. So we are born exposed to EDCs. And wildlife also are exposed to EDCs as well. Now, my research for about the last 15 years or so has focused on how EDCs affect the developing neuroendocrine systems, and we really haven't heard about neuroendocrine systems yet at this meeting, so just a tiny bit of background. And I study hypothalamic neuroendocrine systems of the brain, and of relevance to what I'm going to be talking about today, during development, the brain is sexually differentiated. So we know that male and female brains are different in every species, including in humans. And the mechanisms by which these happen in large part, probably in most part, are due to effects of hormones that a fetus is exposed to, either from its own gonads, from the mother, from amniotic fluid, from placenta. And those hormones, in many cases, get into the nervous system, and they shape and they sculpt brain development in a manner that differs between the males and females because the timing and the amounts of hormones where the sexes are exposed to differ during development. Those early life effects set up the brain in a way that helps it function for the rest of our lives. So we call those early developmental exposures to hormones, or their disruption by EDCs that I'm going to show you, organizational effects of hormones, and then throughout our life, those circuits mature, and then hormones can continue to exert effects. We call those the activational effects. Now, the hypothalamic neuroendocrine systems that I study control reproduction, stress. The hypothalamus is absolutely critical for a number of complex behaviors, social and sexual behaviors. And another thing about the neuroendocrine systems that's very important to know is that they're incredibly abundant in hormone receptors. And so this really, I think, underscores how sensitive the brain is to natural hormones and to their disruption by EDCs. The work that we're doing in the lab really follows up on the model that we call DOHAD, or the developmental origins of health and disease, which has postulated that events that happen very early in life, especially perturbations, can change the trajectory of our entire life as far as our predisposition for different kinds of disorders or for health, and that includes mental and psychological health. Okay, so turning to our research, I'm going to begin by telling you a story about a class of industrial chemicals called polychlorinated biphenyls. They were synthesized and used in heavy industry, many applications for decades and decades, until it was discovered in the 70s that they were carcinogenic and had other health effects, and so they were eventually banned. However, they were very irresponsibly disposed of, and in many cases were just dumped and got into waterways, got into soil. I got interested because I grew up near the Hudson River in New York, which is heavily contaminated with PCBs. And to show you a little chemistry of PCBs, here's a representative structure. It's a biphenolic compound. It can be chlorinated at any and all of these different positions. So there's many different types of PCBs that determine what their properties are, and we study a class, an industrial compound mixture called Aerochlor 1221, which is known to also have estrogenic effects. It was not designed to be an estrogen, but in fact, it can act on estrogen receptors. And so, oops, there I do. Okay. So we are using this developmental origins model in rats by looking at how exposures to PCBs or vehicles prenatally during a period when the neuroendocrine system is being organized by hormones in a sexually dimorphic manner. We give a very short exposure. We expose our rats just for 2 days during gestation, and then we allow them to be born, and we follow their development. We look at hormones and gene expression and protein expression in the brain, and I'll tell you a little bit later also about effects in future generations. But I just would like to show you a small bit of data, a cross-section of our data very quickly. So these are data looking at the exposed offspring, the F1 generation, that were exposed just twice to low doses of PCBs or vehicle. And we've done a number of different types of assays. I'll show you a little immunohistochemistry. A good example is estrogen receptor alpha in the hypothalamus, which I think you can see at this low magnification. There's much more ER alpha in the hypothalamus of an adult female than an adult male rat. That's also true for, excuse me, for humans in this region. And when we expose animals to EDC prenatally, that sex difference is completely abolished. We see the same thing for another protein involved in hypothalamic function, kisspeptin, which is important for reproduction and for metabolic regulation. And we've looked at a number of other proteins as well. But this gives you a sense of the potency of just a small perturbation or reorganizing the brain in a manner that now has permanently changed expression of proteins in the brain. We've also looked at hundreds and hundreds of gene expression in the hypothalamus. Some work Victoria Topper did when she was my MD Ph.D. student in the lab. And in the female brain, just a few examples of interesting genes to me as a neuroendocrinologist: the androgen receptor, estrogen receptor alpha and beta, dopamine receptor, opioid receptors, that in control animals are shown in white, and then in the EDC animals are shown in blue and pink. And you can see in the females, the vulnerability of gene expression to that prenatal exposure. There are sex differences. In males, we only found one gene that was affected in this particular gene array that we used, and that happened to be PER2. For those of you who are interested in circadian rhythms, we think that there may be rhythmic disturbances caused by EDCs. We've also done a tremendous amount of behavioral characterization, and I'm actually just going to show you one behavior because it's interesting and people don't look at it very often, but I think it's a great behavior to look at because it's very naturalistic, and that is ultrasonic vocalizations. So rats vocalize to communicate a lot of different types of information, hedonic, stress, social, sexual interest, and so on. And we recorded ultrasonic vocalizations in our male and female rats that had prenatally been exposed to EDCs. I can play a couple of them. We have flat calls. If you can press the top one, there's no frequency change. Those are thought to be very sort of basic communications. And if you look over here on the right, numbers of those calls in females and in males and vehicle and treated animals, there really was no effect of the EDCs. But we also looked at another type called rise, if you could play that one. So you can hear the upward deflection. Those are thought to be more hedonic. And as you can see here on the right, the males have significantly more of those calls and other types of calls as well that I'm not showing you, whereas these females were not affected for that call type. So to summarize this little cross-section of data and much published data, there is a network of specific genes and proteins in the hypothalamus and really a broader network of regions as well that I haven't talked about that are reprogrammed by prenatal EDCs. Many behaviors are perturbed in a sexually dimorphic manner, and the results really differ depending upon whether you're looking at males or females. Moving on, I wanted to spend my last few minutes talking about the multi-generational effects of EDCs. So consider when we expose a pregnant rat, or when a woman is exposed when she's pregnant, we're exposing multiple generations at the same time. So the mother, her body and her brain is exposed. Her developing fetus, the F1 generation that I just talked about, is exposed. In the F1 generation, the germ cells are developing. Those germ cells, if they go on to mature and be fertilized, are going to become the F2 generation. So we actually expose 3 generations in this type of a model and in the real world. And so it's only at the level of the F3, or the great-grandchild, that there's actually no direct exposure. You could argue how that F2 germline exposure is really an exposure, but as I'll show, as we know, there are effects of EDCs on all of these generations. And what I'm going to talk about is, along with the PCBs I talked about before, we're also looking at a fungicide called Vinclozolin. It is known to block androgen receptors, so it's a known endocrine disruptor. It's used in the grape industry, so when you drink red wine, there's often a little Vinclozolin. And if you play golf, it's very heavily used in the treatment of SOD. And so there's a story behind this. About 12 years ago, Mike Skinner at Washington State made the discovery that if you exposed a pregnant rat to high doses of Vinclozolin, her offspring would develop endocrine diseases, but they accidentally bred the males. And when they started looking at further generations, they discovered that the F2 and the F3 generations were also developing disease through this paternal lineage. And so my partner and spouse, David Cruz, who collaborates with me and I, got animals that were descended from ancestors that had been treated either with Vinclozolin or vehicle, and we put them into a mate preference test to see whether their behavior and whether their attractiveness would be affected. And so I'm going to play 2 videos. There's some background noise, but just ignore the background noise. The experimental setup is, on the left is the female descended from Vinclozolin, a female descended from vehicle, and a male is given a choice between them to see if he can differentiate them and show any preference. And on the right, if you play that video, we also look at how they're interacting, how they're touching noses, et cetera. And what we found was that if males were given a choice between two descendants of Vinclozolin or vehicle, they actually didn't care. Males, like all females, are not very choosy. You know, we can anthropomorphize that as much as we want. However, I wouldn't tell you this story if it weren't interesting, and when females were given a choice, they had a profound preference for the vehicle-descended animal over the Vinclozolin-descended animal. And just for the sake of time, I won't go through the slide, but the paper was published about 10 years ago, and it was an amazing discovery. So we wanted to follow up and try to understand, how does information get programmed in these earlier generations and get transmitted to an F3 generation that had no exposure? And really the two choices, and they're not mutually exclusive, is that either there's some sort of germline programming, and we do think germline programming is playing a role in this, and I'm going to show you some DNA methylation data to support that. The context also needs to be considered, because you may be exposed. But if there are other changes like maternal care or other environmental changes, you can continue to get a phenotype. And we're doing some work right now to try to look at those interactions. So what I'm going to show you in the last couple of slides is some work looking at promoter methylation, because we believe that DNA methylation of sperm may be imparting some of that information. And so Ross Gillette, who just got his Ph.D. a couple months ago, conducted reduced representation bisulfite sequencing of sperm and two brain regions, CA3 of the hippocampus and the central amygdala, based on some behavioral work that we had done showing that there were some behavioral deficits related to the functions of those regions. And I'm just going to quickly go through the sequencing. What he found was that in both generations and with both EDCs, we saw promoter hypermethylation. So these are heat maps of methylation in sperm relative to vehicle in the F1 and F3 generation. We found in every group about several hundred genes that were differentially methylated, mostly hypermethylated. And we saw a very similar thing in the brain, so I just have central amygdala, but we had similar patterns in the hippocampus. And when we compared tissues and tried to look for common genes that were in brain and sperm and were conserved across generations, there actually was a small subset of genes that overlapped, and those were primarily promoter-associated RNAs. So these are RNAs that silence ribosomal RNAs, so they're very important for translation, and they are hypermethylated in the brain and in the sperm of PCB and Vinclozolin males. And so to me, this is an entirely new direction to look for how this information can get conveyed across generations, both to germ cells as well as into somatic tissues, potentially underlying some of the behavioral and other disease states that we see. Okay, so I'm out of time, but this is kind of our view right now, is getting from the sperm to the brain. Work that we're doing right now is trying to get at that, and we're just now starting to do some work in females. So let me end here and thank the funding sources, especially NIEHS, the wonderful people in the lab. Here was the lab about a year ago, our rats, and thank you again for this opportunity to speak to you today.
We have a minute or two for questions. I think someone's coming. Please. Rick.
Yes, Andrea, you showed some really clear effects of the EDCs. Can you put it in perspective a little bit with the doses the humans are exposed to?
Yes. So in the studies that we're doing here, we expose them for very brief periods of time to a dose of 1 milligram per kilogram given to the dams. So our work is the lower dose. I neglected to mention the dose that we used, but it was 1 milligram per kilogram. It probably, people who have looked at concentrations in tissue, including brain tissue, think that it's probably pretty comparable to what's in humans. And we should also keep in mind with humans, we're exposed to so many things throughout our lives. Our rats are probably not being exposed to as much in the lab as humans are throughout their lifetimes. So I think it's a pretty good low-dose model.
I apologize if I've asked you this before, because I know every time I see you talk, I get very disturbed by this. But other than
hopefully not by the research.
No, just thinking about all these things in our environment. Other than avoiding golf courses and red wine, what do you think is the most important thing that pregnant moms can do to prevent exposure in their babies?
Yeah, so I didn't talk about plastics, and I loved hearing Hal's talk yesterday, where he was talking about like making alternatives to plastics. Don't microwave in plastics, don't heat in plastics. That's one of the #1 problems with exposures, is leaching from plastics and food containers and water bottles into what you're actually eating. Wash fruit and vegetables really, really well. That gets rid of most of the pesticides, many of which are also EDCs. If you can eat organic and can afford to do that, then that's a good way to go also.
Okay, wonderful. Thank you very much.